You'll talk more about the abundance of left-handed alpha helices versus right-handed alpha helices in, say, human proteins at another time, as in much higher grade levels. Okay, so here, what happens when you have these secondary structures and you need to put them into the context of a whole three-dimensional polypeptide? Well, then you have tertiary structure. And this depends on amino acid side chains such as ionic bonds, disulfide bridges. Disulfide bridges are by far the strongest of these interactions because a covalent bond is like welding two chunks of a molecule together. And this is one long molecule because the backbone is made of covalent bonds. Now, hydrogen bonds, well, they're stronger than hydrophobic interactions, unless vastly outnumbered, of course. And then there's ionic bonds, which are, they're stronger than hydrogen bonds because they are full charges interacting. But they are, of course, considerably weaker than disulfide bridges, which are formed between cysteine residues. Now, tertiary structure is not the be-all and end-all. We can have multiple polypeptides interacting together to form a functional protein. And this is quaternary structure. You find this in a lot of enzyme complexes, and you also find this in, say, hemoglobin. So hemoglobin has two alpha chains and two beta chains. Each of them contains a heme group or a heme ring. Heme is a very special molecule. It contains an iron atom at the center, or other metal, that works too. And it makes sure that the iron is capable of bonding to oxygen. Now, proteins in solution, they tend to be globular. Why? Well, it's because if you have a lot of hydrophobic residues, they will tend to clump together, yes, instead of be exposed to the water outside. This is due to energy levels. It's just unfavorable thermodynamically to have a whole bunch of hydrophobic residues sticking out. You can find this general phenomenon by, say, adding a drop of oil into water. Can you? Well, not quite. Why? Yeah, okay, small oil droplets floating on water tend to form circles. But because water is so much denser than oil, the oil tends to float to the surface. On the other hand, if you take two liquids that are not mixable, so immiscible, and they have similar density, and then you try to mix them, you will find that you will form droplets, spherical droplets, because that's the least amount of surface area exposed to, well, for the nonpolar molecules, exposed to polar molecules, versus polar exposed to nonpolar surroundings. Okay, so the active sites of enzymes are found on the surface of these globules. Long linear proteins with cross-linking generate the fibers that you find in, say, silk. Fibroin is the main structural protein present. Do not confuse this with fibrin, which is used in blood clotting. And you can also find strong fibers in, say, collagen and keratin. Now, the specific structures are details that we won't cover for now, but they are long and linear, and they have cross-linking. Does this sound familiar in any way? Cross-linking. When we talk about vulcanized rubber, what essentially happens in vulcanized rubber, I won't bother with the actual zigzag patterns. What essentially happens when you vulcanize rubber is that you add sulfur and you form crosslinks. Unvulcanized rubber, natural rubber, is a sticky, gummy liquid, but it's not that hard to rip apart. Whereas ripping apart an eraser without cutting first and then applying leverage is a chore. It is not very easy to get a good enough grip to rip an eraser in half, presuming that it is a reasonably large eraser, of course. So that's because the more you vulcanize it, the more sulfur bonds you form. So say disulfide bridges. What happens when we form lots of disulfide bridges between different polypeptide chains? We end up with a long, linear, and pretty strong protein. We do point out that billiard balls, so pool table balls, they are made generally with vulcanized rubber. In fact, that was one of the first major uses of sufficiently vulcanized rubber, as in add so much sulfur that it acts like ivory. Because, well, okay, billiard balls used to be made out of ivory. But then the game started not being so confined to the upper classes. And ivory was expensive, so they had to find a substitute. Okay. Now, nucleic acids. Nucleic acids are made of subunits called nucleotides. They have a 5-carbon sugar, a nitrogenous base, in this case adenine, and 1 to 3 phosphate groups. Now, we've probably dealt with adenosine triphosphate before, yeah? ATP is the main energy carrier molecule for most organisms. Now, yes, you also find GTP, CTP, and TTP in various roles, most obvious of which is, of course, synthesizing nucleic acids. So if you're making DNA, you had better have plenty of the appropriate triphosphate around. But the nucleoside here, hey, we can have OH in ribose and H in deoxyribose. So when we're synthesizing DNA, we need to get the deoxyribose version of our usual ATP. The usual ATP we deal with has ribose, which means that it is suited for making RNA. It is very important to distinguish between these two forms because DNA needs to be more stable than RNA, and that calls for deoxyribose. So here we notice that the carbons and nitrogens of the base ring structure on the nitrogenous base are labeled 1 to 9, and that the sugar has 1', 2', 3', 4', and 5'. Now, when we talk about synthesis of DNA, we talk about 5' and 3'. That means the directions the sugars are oriented in, because, well, the bases are different, but the sugars, well, they're for the same nucleic acid gonna be the same. And so you can use 5' to 3' or 3' to 5' as indicators of direction. The phosphate groups, well, when you're synthesizing DNA, you end up using only one phosphate. Where do the other two go? Well, they just get broken off as what we call pyrophosphate, and this yields energy. This gives energy to make sure that the bases are correctly matched. And it is an important part of why nucleic acid synthesis is actually reasonably reliable. Reasonably, given the size of the nucleic acid molecules we're replicating, because the error rate is pretty low. Given that we have so many cells and yet cancer generally takes quite a while to develop, yeah, the mutation rate is not generally that high. Okay, so here we have the nucleotides displayed. You might notice that there are 5 of them. We know that, okay, there are 4 nucleotides used for DNA and 4 used for RNA. Uracil is RNA only. It is replaced with thymine in DNA. So adenine, guanine, and cytosine get used in both. C is paired with G. So the purines are these two. So 2 rings. And pyrimidines are these. Maybe it would have been a good idea to put a box around this first. Oh well. So pyrimidines, well, they have a single ring and purines have 2. We should note that the CG bond is stronger than the AT bond in DNA. It has 3 hydrogen bonds between these two, whereas AT only has 2 hydrogen bonds. What does this do? Hey, stronger bond means harder to disrupt, right? So when we heat this up, it takes a higher temperature to make the DNA melt, as in break apart, denature. So the strands separate. What do you think happens to the DNA of organisms that are specialized, or evolved rather, to survive at high temperatures? Well, they have a lot of CG content compared to AT content, as compared to organisms that live at lower temperatures. So a phosphodiester bond is this. Now you notice something here. Hey, even though we know that DNA is synthesized from 5' to 3' as described in textbooks, these are anti-parallel. They do not go in the same direction. Why does 3' to 5' not work so well? Let's see. Okay, the phosphate's sticking out here. Now we have to jam this new sugar in. Now that's not convenient, right? Better to have the sugar here and then just click, click, click. As opposed to, okay guys, this phosphate is sticking out here, this sugar is over here, now we have to jam another sugar in with the other molecule running right here. No. It would be like, oh, instead of stacking chairs on top of one another, we decide that we lay them down and then stack them like that. Yeah, no. You are probably not going to decide to take a bunch of chairs and then shove them onto each other like this, right? No. In this case, it would be nice if we could just, stack the chairs normally. But the problem is the other molecule that we're using for reference is right here. So we have to make these match up. And that means it's much easier to add onto this end instead. Yeah? Okay. So that's why we synthesize from 5' to 3', much to the irritation of, well, a lot of people. Because 3' to 5' would be numerically much easier to deal with, yeah? Except for the fact that reality kind of disagrees. Okay. So, DNA, what's the difference? Well, in DNA, we notice that unlike RNA, we have H here instead of OH. That's why it's deoxyribonucleic acid. RNA can actually also do anti-parallel base bonding. It's just that the structure is less stable due to this OH. Hence why DNA ended up being the genetic code of life, while RNA still acts as an intermediary. What's another difference between these two? Uracil, instead of thymine, is used in RNA. Now, carbohydrates, lipids, proteins, and nucleic acids, obviously they all have their important roles. They also interact with each other to a great degree, and they sometimes overlap. For example, glycoproteins. That's literally sugar and proteins. I wonder what the structure could possibly involve, right? Okay, now proteins and nucleic acids. Guys, chromosomes are made of proteins and nucleic acids. The proteins are histones, and they also have transcription factors attached to various binding sites, and okay, some other stuff. Okay, what's the actual genetic material? Well, when people first investigated, they thought protein was the genetic material. Why? Because the nucleic acids only had 4 bases. Hey, that looks too simple. Yeah, this was in the age of Morse code transmissions, so why 4 bases is too simple for the genetic code, and dot dash is apparently not too simple for human communications. Well, they figured out that it was different a little later. Okay, now for lipids and proteins. Yeah, these also have some overlap. So, in fact, do carbohydrates and lipids. They can attach groups onto each other to perform novel functions. So all of these are important aspects of life. Amino acids are the monomers that make up proteins. There are 20 different amino acids that are commonly used by life on Earth. There are a few other amino acids that are used by a few life forms. And even humans technically also use one more amino acid, and it is called selenocysteine. It replaces the sulfur in cysteine with selenium. This is why selenium is important to human health. But too much selenium is toxic, so please be careful. We should note that Brazil nuts actually contain a very large amount of selenium, to the point where eating too many is not very good for you. They also happen to contain radium because of just how deep their roots grow, but that's another story. Okay, so the amino acids are so named because they contain an amino group and a carboxylic acid group, amino acid. And they contain an R group. What is this R group? It's rest of molecule, remainder, or residue, depending on who you ask. And the R group is what gives each amino acid its unique characteristics. Proteins are complex polymers. They have primary structure, where it's just, sequence of amino acids from N terminus to C terminus. They have secondary structure, so hey, what happens when this chain of amino acids spontaneously folds to produce a alpha helix or beta pleated sheet, or any other structure for that matter? Now, they also have tertiary structure, which is what happens when you take the secondary structure pieces and you perhaps weld them together with disulfide bridges, perhaps you use ionic bonding, or perhaps just hydrophobic bonding, as in the interior of many globular proteins. Why the interior? Well, okay, when you put a protein in an aqueous solution, it will generally rearrange such that the hydrophobic parts point inward, so on the inside, and the hydrophilic amino acids point outward. This is energetically favorable. Now, of course, there are circumstances where the hydrophobic groups point outward. This is actually an identifying characteristic when you're looking at a protein that passes through the plasma membrane, so the cell membrane, or perhaps some other membrane. Any phospholipid membrane will end up showing you a sequence of amino acids inside for a protein that are hydrophobic. If you need to map out a protein that crosses the membrane several times, well, just take the hydrophobicity index of the amino acid residues, and you will be able to tell which spans of the protein cross that membrane. Now then there's quaternary structure, which was what happens when you take multiple polypeptides and make them interact somehow. whether by, okay, spatially squishing them together and watching the hydrophobic bonds, or for that matter, ionic bonding work, or what else could you do? You could potentially use disulfide bridges with multiple polypeptides, and it'll still weld them together into one big molecule, or several big molecules. So when you take these complex polymers and give them additional levels of structure, you end up with emergent properties that any one previous level might not have had. This is an important theme of life, the theme of emergent properties. So the whole is greater than the sum of its parts. A pile of pieces does not result in something you can pedal down the street, even if these are the disassembled parts of a bicycle. Well, does not result in it unless you're willing to put in a lot of work, but that's another story. Okay, so some proteins, they need non-protein components called prosthetic groups to function properly. This often means, okay guys, time to weld a sugar on here or over there or wherever. All nucleotides, well, they consist of 3 parts: a nitrogenous base, a 5-carbon sugar, and 1 to 3 phosphates. Nucleotides make up DNA, deoxyribonucleic acid, and RNA, so ribonucleic acid. Now, we should note that nucleic acids are also capable of catalyzing reactions, like proteins are capable of catalyzing reactions. Now, yes, okay, the range of reactions they can catalyze differ, but that's something interesting that we should know. Now, the 2 general types of nitrogenous bases are pyrimidines, uracil, thymine, and cytosine, and purines, so adenine and guanine. Now, uracil is only found in RNA and thymine only in DNA. They bind to adenine with 2 hydrogen bonds. Cytosine and guanine have 3, which makes their bond stronger. And thus, the DNA of organisms that live at high temperatures have been selected to have more CG bonds than AT bonds, compared to organisms that live at lower temperatures. Okay, that's it for this section. In the next section, we can talk about what lets all of these parts, all of these various molecules work together. Well, some reactions have to happen, right? But we know that a lot of the reactions, if we let them happen naturally, are kind of slow. If you let starch just sit there with nothing helping it along, it takes an eternity for it to just spontaneously turn into simple sugars. Obviously, we don't have an eternity to wait for living organisms, right? So, we need to use enzymes. Currently, enzymes, we generally deal with mostly proteins. But we should note that nucleic acids can catalyze some reactions. In fact, ribosomal RNA is important in terms of creating proteins. Okay, so that's it for this section, and we'll see each other again next section.